Right. Hello, so welcome to my hot wash video. So my video is going to be about snares and neurotransmission specifically. So introduction to snares. What are snares? So this is what it stands for. It's soluble NSF attachment protein receptor. So that's quite a mouthful, which is why we call them snares. And what they are is they're proteins, specifically alpha helices, and they're embedded in plasma membranes. And they aid in the release of vesicle contents to the outside of the cell. So they help with exocytosis. And specifically, they're really well known for helping with neurotransmission. So it's going to release neurotransmitters outside of the cell. So both the vesicles and target membranes are going to have snares, and those are going to be called the V snares and the T snares. And they're going to be complementary to each other so that the vesicles get released in the right place, because they'll be able to recognize each other and bind to the right place. So when they recognize each other, they combine to form this alpha helix bundle complex, and this pulls the two membranes close together and preps them for fusion. And then once signaled, in neurotransmitters case, usually that's by a rapid influx of calcium that's going to bind to some chaperone proteins that's going to signal for this fusion to happen. These two membranes are going to fuse, and the contents of the vesicles are going to be released out of the cell. So that's kind of the essence of what it does altogether. So let's go into that a little bit deeper. So what are the first steps? So pre this rapid calcium influx, we're going to do a few key steps. And that's tethering, docking, priming. So there's already going to be some low concentrations of calcium already in the cell. And that's going to promote movement of the synaptic vesicles towards the membranes and some calcium ion gates to prepare it for fusion. So tethering, they're going to recognize each other, the V snares and the T snares. Docking, they're actually going to start to form this complex known as the snare pin complex. And then priming, these chaperone proteins are going to come down and clamp down and kind of anchor them down and really promote, kind of the stress and the rearrangement of the membranes to prepare for fusion. And then at that point, they're just waiting for the major calcium influx that's going to allow the fusion to happen. So let's take a closer look at that. So here's the tethering, where they kind of recognize each other and start forming the bundle. And then here's the docking, where the bundle actually starts to form. And as you can see, it pulls the two membranes really close together. For example, it started here at 8 nanometers apart. Once the snare pin complex is there, they're only 2 nanometers apart, which is really important. And then this is just a closer look at what that alpha helix bundle looks like. It's going to be 4 alpha helices, and it's not super symmetrical, it's asymmetrical, and it's going to hold these two really close together. And then here's just another visualization of that, where they recognize each other, and then they clamp down and pull each other really close together. And what are some of the proteins involved? So there are the synaptotagmins and the complexins. And the synaptotagmins are really, really important, and they kind of serve a dual purpose. So for one, they're going to clamp down on the snare pin to bring the membranes closer and create stress on the membranes to promote fusion. And that's going to be this purple here and this brown-gray here. So that's before this calcium influx. Once the calcium influx happens, this is what makes the synaptotagmins really important. They're actually what's going to bind to the calcium and stimulate the fusion. So they're the receptors of that calcium signal that helps the snares fuse together and actually release the neurotransmitters. And then the other important protein are the complexins. And these are involved in docking the V snares to the T snares, and they help ease repulsion between the membranes in order to stabilize them. Now it's going to be this yellow here and this orange here. So they're just going to help further to bring the two membranes close together. Excuse me. And then note, synaptotagmins are partially in effect during the tethering priming stages, but are waiting for the influx of calcium to fully snap into work, which I kind of mentioned. And you will see that right here. So as you can see here, and then as soon as it binds to the calcium, that's what's going to trigger the release that happens here. So it's kind of this, I'll just show you here because I think this is the clearest way to show what happens. So they're going to start tethering and docking, and bam, they have the snare pin. And then the calcium's going to come and releases everything, which is really cool. and then it's going to dissociate. And I didn't learn too much about this, but I did learn that this is ATP driven. Like, you need ATP hydrolysis in order to release all the snare pins from each other, because it's a pretty stable complex that gets formed. So that's basically the essence of it. Phew. Abnormalities. So what happens when there's some irregularities with the snares? So this is not going to be good, because the snares are really vital to sending these signals. Like, they actually are what lets these signals be released. So what does deficiency cause? So it prevents cell communication, and it damages motor control, because snares help your muscles and your cardiac muscles move, and also leads to neural damage. That's mostly what a lot of the papers I read focused on, was the neural damage aspect of this. So it can contribute to Huntington's disease, Alzheimer's, Parkinson's, and ALS. It's been seen like a deficiency of snare pins, or snares in general, has been associated with schizophrenia. It's been associated with autism and learning impairments. And then overexpression, which is too much snares, it's been seen in some patients with bipolar disorder, which is really interesting too. And then here's my sources. And that's basically it. That's how snares work. Thank you.